Hello, everybody. Welcome back. So in our last lecture, we were discussing really the chemical structure of DNA and how DNA essentially is held together by these important bonds between adenine, thymine, cytosine, and guanine, but also these important phosphate sugar bonds that make up the backbone of the DNA. And so now what we're going to be discussing is we're going to be sort of, again, zooming out a little bit to understand how scientists have been able to basically harness the diversity of DNA in a lab environment and use it for forensics and a myriad of other uses. But your book this week focuses on the Innocence Project and understanding how scientists have been able to use DNA to go back and evaluate cases from the past that have where DNA evidence wasn't used previously. So, in order to understand this, we have to understand the fact that the specific sequence of nucleotides, those A, T, C, G, adenine, thymine, cytosine, and guanine groups along the strands of DNA is unique to each individual, save for identical twins. And we can actually extract DNA from cells to create an individual's DNA profile, so understanding what makes up their DNA. And we can basically get a visual representation of a person's DNA sequence. And in order to get this whole profile and this whole genome, you need sometimes many copies of their DNA. So knowing these two facts, how do biologists make use of this information to actually understand the unique identity of an individual using DNA? And the secret really lies in the process of DNA replication, which is something we're going to be going into more specifically next week. But the important things to take away for this lecture is to understand that this is just the natural process by which cells make an identical copy of a DNA molecule. And essentially, this process takes advantage of those complementary base pairing rules, where adenine must always pair with a thymine and guanine must always pair with a cytosine. So let's look at this sort of glossy picture from your book and understanding how cells reproduce. So when cells reproduce, they must first replicate their DNA, so then that each of those new cells contain a copy of the original DNA molecule. And so what happens essentially when we have this, let's look at this region of DNA, the hydrogen bonds holding those base pairs are broken apart, unwound, and then after a replication, each of the new DNA will have a twist of the original strand on one side and a twist of basically this newly replicated strand. And the way this happens is essentially DNA is unwound, and these base pairs are broken, and an enzyme, a very important enzyme called DNA polymerase, again, we know it's an enzyme because of this ACE ending, DNA polymerase, goes through and basically reads the bonds that are left over on this one side of DNA and adds the complementary nucleotides. In this case, we're seeing green. So previously, for example, we had an AC pair. The DNA polymerase adds the corresponding TG pair. And that's how we get sort of this now complete strand of DNA that includes blue, the old strand of DNA, and then this new strand of DNA. So there are a couple of broad steps that are important in this. The first thing to note when we're talking about DNA replication is first thing that happens is these bonds are broken and that helicase is unwound from those very discrete particles. Then there is the replication phase, and this is where the DNA polymerase comes in and adds those adjoining base pairs. So you get one strand of the original DNA and then one strand of the new nucleotides added. So when we're talking about sort of new and old DNA, quote unquote, we're talking really about this semi-conservative mechanism. So because we're producing two copies of the original DNA molecule, but we're also using the original molecule as well. So it's called a semi-conserved mechanism, where, again, each molecule is going to have one strand of the original DNA and one strand of the newly created DNA from those, again, unique base pairings. So how can we take advantage of this DNA replication strategy and actually use it in a laboratory context? So I'm going to be talking about probably the most important biological bench science of the polymerase chain reaction, or PCR. So this is how we're able to start creating a DNA profile. But at its base, PCR is basically a laboratory technique that is used to replicate and then amplify a specific segment of a DNA. And in order to replicate and amplify these specific units, we need a couple things to go in to this test tube to sort of start creating this process. First, we need DNA from a sample. We need the distinct nucleotides, A, T, C, and G. So we have all of those distinct nucleotides that are going to be floating around in this. We need the DNA polymerase, so that important enzyme that then takes those nucleotides and puts them onto the new strand of DNA. And then primers. And primers are simply short segments of DNA that guide the DNA polymerase to the section of the DNA to copy. And I think we're going to go ahead and end that there for today, and we're going to pick up where we're going to specifically look at how this polymerase chain reaction occurs. Okay, see you soon.